And for a lot of stress-related disorders, including PTSD, these are two of the key features. So this was all work that was kind of already out there when I started my postdoc work in this field. And all we had to do was to say, okay, is this going to translate to humans? Seems pretty straightforward. But so what I'll do today is I'll kind of go through like the 3 steps that we take in doing this work. So I'll talk a little bit about what happens when we modulate endocannabinoid tone in healthy people. Then we'll look into clinical populations, taking a cross-sectional approach and see, do we see dysregulation of endocannabinoid function in clinical populations? And then I'll talk a bit about, our attempts to pharmacologically enhance endocannabinoid tone to improve treatment outcomes in a clinical population. And I'll go through the studies in the healthy humans a bit quickly because it's published data. So if you're interested in it, you can kind of dig into it. But I will talk a bit about some of the structure of the tasks because we use those repeatedly. So the first thing we wanted to do was to see, what are the consequences of having more anandamide in healthy people? And interestingly, there is a SNP within the FA gene. And if you're a carrier of this variant A allele, what happens is you encode a FA protein that's 30% less active. So it's not quite as good as its job in chewing up anandamide. So these individuals have more anandamide at baseline. So here we have people who kind of already exist that are walking around with higher anandamide, and we can say, what are the behavioral consequences of that? But from a therapeutic standpoint, that's not super helpful because when you do these behavioral genetic studies, we know these individuals had high anandamide their whole life. So maybe it was something that was throughout development that's driving these effects. We had to be sure that we could recapitulate these effects using kind of acute pharmacological intervention. So we followed up this study with a pharmacological proof of concept where we were able to get our hands on one of those FA inhibitors, again, that aren't currently approved for anything. And we were able to see what happens if you give these drugs to healthy people. So I'm going to show this data kind of side by side. So these studies were run consecutively. We did the genetics and then the pharmacology, but I'm going to show them side by side just to compare and contrast the effects. So in study 1, again, this is this behavioral genetics approach. And we weren't the first to publish on this specific variant. But the one thing that we did that was unique at the time was we recruited a whole group of these AA individuals. So people that are homozygous for that less active FA protein, they're very rare. It's about 5% of the population that are homozygous. So we had to screen almost 450 people just to get these 25 individuals. And we're glad we did because we did see some kind of unique characteristics specifically in these individuals. For study 2, these are the people who were in the drug treatment group, or in the drug treatment study. So we had people who were randomized either to a placebo or the FA inhibitor. It was a Pfizer molecule. Now Jazz Pharmaceuticals has it. And the methods are exactly the same across studies, with the slight difference that in the genetic study, this was just day 1 and 2 of their participation. In the drug study, our individuals were on the drug for 10 days in total. And that's just to get steady state elevations in anandamide. So there's testing days were on days 9 and 10 of dosing. And the first thing we wanted to look at was, do we actually see higher anandamide in these individuals? So we, these being humans, we can't look in the brain or do much really cool invasive stuff, but we can measure endocannabinoids peripherally. And what we do see is with this kind of gene dose dependent increase in anandamide levels. So the more A alleles you have, the more anandamide you have, which makes sense. It's not a huge effect. Again, that A allele only encodes for kind of a 30% reduction in FA activity. So it's small, but again, these people have had this higher anandamide their whole life. Whereas in the drug treatment group, we get this huge elevation in anandamide. This is actually a log scale because it wouldn't fit otherwise. So this is a really huge increase in anandamide, much greater than kind of physiologically normal levels. But again, it's only during that short period when they're on drug. And then we also show that we are increasing anandamide and we're not changing anything in 2-AG, which is the other major endocannabinoid ligand. It's not regulated by FA activity, so we shouldn't be messing with it. And we see that that's the case. So one of the tasks we put our participants through is this acute stress reactivity task. So they don't really know what's going to happen, but they're told, okay, you have to put your hand in this ice cold water, and then a buzzer is going to go off, and then we give them math problems to do. And if they're wrong, we tell them that they're wrong and they have to start over. And if they're doing it right, we tell them they have to hurry up and go faster. And then another buzzer goes off and they have to put their hand back in the cold water. And they don't know how long it's going to take, but it's a 10-minute task. And what we do is we collect blood samples before and after, we collect psychophysiological recordings, and then we also get subjective reports of how stressful they say it was. And what we find here, we're looking at skin conductance, so kind of autonomic stress reactivity. In our genetic study, there's no effect of that genotype on skin conductance. We do see in the drug treatment group, though, that we see individuals that are given the FAAH inhibitor, again, who have high anandamide, are showing less of a physiological stress response. We also see a small but significant decrease in how stressful they say the task is, again, only in the drug treatment group, not in the genetic study. But we don't see any differences in their peak cortisol response. So the cortisol reaction to stress is the same, but we do see that in the drug treatment group, we have reductions in autonomic and self-reported stress reactivity. So only in the pharmacological task do we see these effects. And we think that it might just be that in the pharmacological study, people had such high levels of anandamide that maybe, you know, that small increase you see with the genetic study isn't enough to have these stress attenuating effects. We also look at emotion processing in these individuals, and we do ask them questions about how they feel, but we also record from their facial muscles. We'll show them these normatively rated positive, neutral, and negative images. We'll record their facial muscle reactions to them, and then ask them how those pictures made them feel. And here I'm showing you the corrugator muscle that's activated when you frown. And you can see here when we show individuals our kind of negative images, you get an increase in the corrugator muscle. When you show them positive images, you get a relaxation of the corrugator muscle, but we don't see any differences between the genotype groups or the drug treatment groups. So there's no difference in their baseline emotion processing. But then what we did is we had them do this task again after they did that stressor I just talked about. And our rationale for that was, again, if you remember back to the animal literature I mentioned, we didn't see any effect of the FAAH inhibitor at baseline, but when they turned on that bright overhead light and made it particularly stressful, that's when we saw an effect of the FAAH inhibitor. So we decided to do this task again after we stressed them out with the cold water math problem task. And here we're looking at the change in corrugator reactivity due to stress. So and overall, you see kind of everything's a little bit worse after stress, except for in our AA individuals. Again, those are the ones that have high anandamide. They're actually showing less negative responses to negative images following stress exposure. So bad things are a little bit less bad after stress for them. And we were able to replicate this effect with our drug treatment group. So if we pharmacologically enhance anandamide, we also see that we show less negative emotion following stress exposure. Which we thought this was interesting, but then it begs the question, you know, why do we see differences at this time point and not at that baseline time point where, you know, I told you there was no differences between groups. So what's happening, you know, during that stress exposure? And one potentially obvious place to look, it wasn't so obvious to us at the beginning, but was actually at anandamide levels. So what's anandamide doing during this period? And you can see here, we have our stress or our control task. And then we get this kind of transient increase in anandamide. And then after the stress task, you get this kind of crash of anandamide. And it's during this recovery period where they're doing that second emotion processing task. So where those differences were emerging. So when we looked at our genetic set and we broke it down by group, what we thought was pretty interesting was that during this recovery period, what you're seeing is that everyone's showing this decrease in anandamide levels following stress, except for those AA individuals. Again, these are those very rare individuals who have really high anandamide to begin with, and they're not showing any stress-induced decreases in anandamide. And we thought that was interesting, but we didn't really know what to make of it because at the time, there wasn't a whole lot of literature out there on these more dynamic responses of endocannabinoids in humans. And luckily, I was going to a conference where I got a travel award and I got to pick a mentor. So I picked Matt Hill because he was like the anandamide guy at the time, still is. And I wanted to show him this data to see if he thought that it was something that was interesting or not. And it turned out that Matt had been using this FA mouse. So he's using a mouse model where that FA SNP was actually put in. And he was doing the same tests where he was stressing animals and seeing what happened to anandamide. And just like what we saw was, you get this decrease in anandamide levels in general here in the amygdala, but the AA animals don't show that stress-induced decrease in anandamide. And he saw that in the amygdala and in the prefrontal cortex. And then after we met up and we showed each other the data, I asked if he could go back to look in the periphery because that's all we can measure in humans, and animal researchers like forget that there's just regular blood around. They're interested in like these specific brain regions, but we want to know if these peripheral measures really mattered or were at all related. And we did see the same thing in the animals, where even in the periphery, we don't see that stress-induced decrease in those AA individuals. So this was, really exciting for us. We ended up putting the data together and publishing just a few months later. And it was really encouraging because here we had, not only did we replicate an effect, but we replicated effect across species, doing these studies without the other even knowing about it. So to summarize these studies that we did in healthy people, we see that when we have elevated anandamide, it can promote the regulation of negative emotion. So that was in both studies, as well as facilitate fear extinction, which I didn't mention. But once we look at stress reactivity, we found that only when we pharmacologically enhance anandamide do we see these beneficial effects. And again, we think that's just because when you use the drug to increase anandamide, you get such high levels of anandamide that wouldn't be, physiologically possible otherwise. So this made us think that, maybe anandamide is relevant to doing something that's useful. Should we actually try to target this therapeutically? But before we did that, we thought, well, let's see if we see any dysregulation of endocannabinoid function in clinical individuals. And here, this data came from a study where we're looking at people who were exposed to trauma during childhood, and then whether or not they were going to have an increased risk for developing a substance use disorder as adults. And this is another paper that's published, led by Andrea Capucin, who's like a just really brilliant physician scientist back in Sweden. And she used this prospective approach where we have individuals with documented childhood trauma, and then we could follow them up and to see if they had any contact with our substance dependence clinics. And what she finds, not surprisingly, is that, yeah, you do see this huge increase in the risk for developing a substance use disorder if you were exposed to trauma during childhood. But then the question became, what is it? What are the mechanisms of that? And in order to start looking at these mechanisms, we actually invited some of these individuals into the lab. So we were able to invite in a group of people who had, documented childhood trauma as children or adolescents, and then documented substance use disorder issues. Then we invited individuals who had documented childhood trauma but did not develop a substance use disorder. People with substance use disorder only, but no documented childhood trauma, and then healthy controls who have no trauma, no substance use disorder history. And we measured endocannabinoid levels in these individuals just at baseline. And here we're looking at 2-AG, so the other endocannabinoid, and we see that all of our trauma-exposed individuals have lower 2-AG as compared to controls. But when we looked at anandamide, we see this trauma-only group actually has really high anandamide levels as compared to controls. So again, these are the people who were exposed to trauma as children, but for some reason did not develop a substance use disorder. So we're saying, maybe they're resilient to developing a substance use disorder because of this high anandamide. And we know that it's specific to the endocannabinoid system because we don't see any difference in cortisol. We also expose these individuals to that same stress task I talked about. And what we find is not only do these individuals have high anandamide to begin with, but it stays high throughout the stress task. So it's high at baseline, it's high throughout stress. And again, it's specific to anandamide. We don't see any group differences in cortisol. So, our thought is, what are the behavioral consequences of having high anandamide in these individuals? We had them do a similar task that I mentioned before, where they're shown positive, neutral, and negative images. So positive pictures of puppies, negative, pictures of vomit. And we asked them how it made them feel on the orthogonal dimensions of valence, so negative to positive, and arousal, so kind of the magnitude of the response that they feel. And what we see for valence ratings, no group differences. Puppies are good, vomit is bad. But when we look at arousal, so again, this is how much of a response do you feel? What we find is that trauma-only group, that resilient group, they report feeling kind of less of a response to negative images than controls. Suggesting that, maybe there's something different in their kind of neural circuitry underlying emotion regulation. And it happens that we actually put these individuals through a neuroimaging session that was led by another brilliant colleague of mine, Karina. So Karina had these individuals do this emotion regulation task where you see a face and it has a word over it, and you have to ignore what the word says and tell us what the emotion of the face is. And what Karina found is that trauma-only group, again, they have high anandamide, they say that negative things elicit less of a response. They're actually showing greater activation in the ventromedial prefrontal cortex, which is a key area we know that's involved in emotion regulation. They also show greater activation in areas in the anterior insula and anterior mid-cingulate cortex. These are areas that are critical kind of in salience processing. So they're seeing kind of greater activation in areas that are important for detecting important information in the environment, but then also regulating their emotional responses to those stimuli. So when we started to kind of put all of this data together from this group, I didn't show you the data, but we have clinical ratings where this trauma plus substance use group has greater depression and anxiety symptoms. We saw that everybody had low TAG, but in this trauma-only group, they had high anandamide, they had greater activation in emotion regulation and salience processing areas, and they felt less reactivity to negative emotional images. So we kind of went into this looking for what might make people vulnerable, because a lot of times in psychiatric research, we want to know kind of why do things go wrong? But we think we might have actually kind of stumbled upon, what makes some individuals resilient? Why were these individuals resilient following their trauma exposure and did not develop a substance use disorder? And we think maybe anandamide might be pointing us in the right direction. The issue with these sorts of cross-sectional studies, though, is we don't know why these people had high anandamide. Were they just born with higher levels of anandamide inherently and thus protected? Or was it something about their traumatic experiences, their repeated stress exposure that increased the anandamide they had? And if so, why some people and not the others? This is stuff we just can't answer in humans. So we have colleagues in Munich who are going back to animal models now and trying to figure out kind of the temporal sequence of these events. But what we can do in humans is actually see if we pharmacologically enhance anandamide, can we improve treatment outcomes? And this is an ongoing study that's what I'm doing in collaboration with Marcus Heilig back in Sweden. Marcus was my postdoc advisor, and we're collaborating on this project because it took us a really long time to get it going. But again, we're looking at FAAH inhibition. We've shown that if you inhibit FAAH, you increase anandamide, you can increase fear extinction, you can reduce stress and reduce negative emotion. These are key features of the symptoms you see in PTSD. So what we're doing is we're recruiting patients with PTSD diagnosis, and they'll be randomized to receive a FAAH inhibitor or placebo, and they'll be taking that drug for 12 weeks. And then we'll be getting their kind of standard clinical assessed outcomes of PTSD symptoms, plus a lot of self-report measures. So measures of, their self-reported symptoms, sleep, anxiety, mood, and a lot of blood samples too. And then after the first 4 weeks, everyone's going to come in and they're going to do the exact same set of tasks that I've talked about today. So stress, motion processing, fear conditioning and extinction, and the same neuroimaging task that Karina had used previously. And then after that, everyone's going to do an 8-week treatment of prolonged exposure therapy. So prolonged exposure therapy is kind of like the gold standard treatment for PTSD. It's based on the idea of kind of extinction learning. So we're collaborating with Gerhard Andersen, who has an online version of this where people are able to kind of get the treatment without having to leave their home. And this was important to us because we started this project during COVID. So we were still able to give people access to this therapy without them having to kind of risk coming into the clinic. So this study is still ongoing, so I unfortunately don't have any results, but we're getting close to the end. So we hope soon we'll know, if we pharmacologically enhance anandamide, are we going to improve treatment outcomes in this PTSD population? And we'll get some mechanistic information as to what's happening with, emotion regulation, fear extinction, and stress reactivity. So just to summarize, I talked a bit about the beneficial effects of elevated anandamide in healthy people. And again, that's all published data. I mentioned, people who have high anandamide that were exposed to trauma previously, that a high anandamide might be kind of protective against developing maladaptive stress coping strategies like substance use disorders. This is data that isn't currently published, but we just resubmitted revision, so hopefully it'll be out soon. We next will hope to see, will patients with PTSD benefit from elevated anandamide? And one thing I want to point out is this is all directly based on animal models. And all of it done in collaboration with all the animal researchers. And I think that's why we wrote this review not long ago where we talked about some of the successes in the cannabinoid field. It's not specific to this anandamide story. It's just my favorite one because I've been working on it for so long. But Christine Rabinack at Wayne State is doing just really pioneering work looking at THC, so activating cannabinoid receptors with exogenous cannabinoids. She's done the same sorts of studies in healthy people. She's also running a PTSD clinical trial combining THC with prolonged exposure therapy. So I think, within the next year or two, we're going to have some really definitive answers as to, can we target the endocannabinoid system for PTSD? And is it kind of how we expected it would be based on the animal literature? So I just want to finish by quickly thanking everyone who's been working on this. So again, I did all of this work in Sweden. So I have a lot of fantastic collaborators there. And then my own group, people who have started the study, who are running it back in Sweden, and those who will help analyze the data next year once we have it. And yeah, I'm happy to take questions.